Hey Team Bio, welcome to your screencast on, this is actually kind of a review of mitosis and an explanation of meiosis. And also kind of applying a lot of the vocabulary that you learned in your last screencast, homologous chromosomes versus sister chromatids. We're gonna kind of bring that all into action here as we discuss the compare and contrast meiosis and mitosis. Okay, so we're going to start out with our starting cell here. This is what we call a diploid cell. A diploid cell has 2 copies of every chromosome. 2 copies of every chromosome. So a copy, one from biological dad and another copy from biological mom. So when we talk about diploid organisms, humans are an example of them. We abbreviate, we call this number I guess we say this would be a cell that is 2n, where n is equal to the number of homologous. It's a G, O-U-S. So humans, n is equal to 23. We have 23 pairs of homologous chromosomes. So a diploid cell of a human is going to have 46 chromosomes total. Okay, so that's a human cell. The cell that we're looking at here, the number of homologous chromosomes is 2. So this chromosome, the diploid number of this cell would be 2 times 2, which is 4. So know that our schematic is that if we were to actually do this with a human, we'd have 23 pairs of homologous chromosomes, 46 in total, but we're working with a simpler model here. Okay, mitosis. Overall goal of mitosis is to divide one cell into 2. So one cell becomes 2 cells. And we want to make exact replica. So these 2 cells, the relationship that they have to one another is they're clones. Okay, the goal of meiosis is twofold. We want to create variation, and we also want to cut genetic info, G-E-N-E, genetic info in half. And the reason for this is that an egg, which is 23 chromosomes, is going to meet with a sperm, which is also 23 chromosomes, and together they're going to make a fertilized egg or an embryo, which has 46. So part of the goal of meiosis is to cut the genetic info in half. If we're starting out with a cell that has 46 chromosomes, we want to end up with some cells that only have 23 pairs of chromosomes. So the whole goal of meiosis is to half the genetic info and to create some variation along the way. Okay, so how does mitosis accomplish this goal of making 2 cells that are clones of one another's, exact copies? Well, we've talked about this in relatively great detail, but you all know that in interphase, the DNA, the cell does its normal cell life self, and then also within interphase, there's this phase called S phase, and the DNA is replicated. PREP is replicated. So we go from a chromosome that looks like this, kind of with a center here, and then it undergoes DNA replication, and it starts to look like this. So the relationship between this arm and this arm of the chromosome, we call these sister chromatids. So this would be during S phase, is this arrow here. It's a horrible arrow. And they have all the same genes. All the same genes and alleles. So they are clones of one another. And once again, this happened during S phase, during DNA replication. And we're going to talk in much more detail about the mechanism of how DNA is replicated in a few weeks. So just hold on to that thought if you have questions about it. Okay, so we get to prophase, and all the DNA looks like little X's. These little X's, once again, are sister chromatids. TI. And so they share all the same genes, same alleles. So this arm of the chromosome is a clone of this arm of the chromosome. Okay, this red chromosome here, let's call this chromosome number 1 from dad. So C number 1 from dad. And this blue chromosome here is chromosome number 1 from mom. The relationship that these chromosomes have to one another is they're called homologous chromosomes. They share, S-H-A-R-E, same genes, but different alleles. And this is going to be important in a bit. So the way the chromosomes line up during metaphase is that all the chromosomes lie on the metaphase plate, and then sister chromatids are pulled apart from one another in anaphase. So remember, this has all the same genes and the same alleles as this one. And this chromosome here, this sister chromatid has all the same genes and alleles as this one, and it has the same genes as this one, but not the same alleles. So anyway, when we divide these two cells, ultimately, we end up with one cell that has a copy of every single chromosome that this original cell had, and this cell also has a copy of every single chromosome that this original cell had. So they end up with the same number of chromosomes in total. So these are both still diploid cells, and the diploid number has not changed. So if this were a human, we would see this cell has 46 chromosomes, and we'd see this cell has 46 chromosomes. Somatic cells is just a fancy way of saying cells of the body. This is not equal to your germ, or I should say your gametes, not equal to gametes. Your gametes are either your egg or your sperm. So somatic cells are all your body cells except for your eggs and sperm. So this is the process by which all your cells grow and divide when they're body cells. Okay, let's compare this to what's happening in meiosis. As we said before, the point is to create some variation and to cut the genetic material into half. So how do we do that? The first thing that's different about meiosis compared to mitosis is you'll notice during prophase of meiosis, we have homologous chromosomes lining up on the metaphase plate. So this is important. If this is happening in you, this is the chromosome that you got from your mom, and this is the chromosome that you got from your dad. Now, they're homologous, so this means that they share all the same genes. So this one, let's say you got this one from dad, and you got this one from mom. They're homologous chromosomes, so they have same genes, but different alleles. And then, so this is a homologous chromosome relative to this. The relationship that these two red guys have to one another is they're sister chromatids. That's way too small. Sorry. Ooh. Campbell's going to be mad at me. So I'm writing way too small. Okay, let's say this one is the one that you got from dad. This is the one that you got from mom. These are sister chromatids. So this arm is exactly the same as this arm. These are clones. This is clone of this. It's not the same as this. This one is the same as this, but these are homologous chromosomes. They share the same genes, but they have different alleles. Okay, so what happens during prophase? They do this, process called crossing over, and this is where the chromosome that you got from your dad crosses over with the chromosome that you got from your mom. They form this structure called chiasma, or if it's plural, it can happen at multiple places along the chromosome, so it's chiasmata, kind of like stoma and stomata, and this can happen anywhere on the chromosome. And when this happens, the chromosome arm that was on the chromosome you got from your mom flops over and exchanges genetic information with the chromosome that you got from your dad. So when you ultimately look at the results, you see that this chromatid is a recombinant. A recombinant chromosome means that part of the DNA was the DNA that you got from your dad, and then part of the DNA was the DNA that you got from your mom. So now, remember, these chromosomes originally had different alleles. And so there was a combination of alleles on this chromosome that was different than the combination of alleles that was on this chromosome, but they had all the same genes. So now you've created a new chromatid here that has all the same genes on it as before, but now this combination of alleles is not the same as this one. So were you to look at this chromatid compared to this chromatid now, they're not clones anymore. This has a new segment of alleles on it. So imagine we had a gene up here that was big A, and a gene down here that was originally big B. Now the big B allele is over here, and maybe there's a little B allele on this side and a little B allele on this side, and this was little a and little a. So this chromatid now has a new combination where the little a allele is matched up with the big B allele, and the big A allele is matched up with the little B allele. If that doesn't make much sense yet, that's okay. But know that this process creates a new combination of alleles, a totally unique combination. Okay, so that's one way in which meiosis creates variation through this crossing over mechanism where homologous chromosomes exchange genetic information. Okay, the next way that meiosis creates variation is through the arrangement of the chromosomes at metaphase. Okay, so there are two equally probable, in this case, arrangements of the chromosomes at metaphase. So we could line up with all the chromosomes that you got from mom on one side of the cell and all the chromosomes that you got from dad on the other side of the cell. And so we see here that ultimately the gametes, we're making this combination, just chromosomes from your mom and just chromosomes from your dad over here. However, this is an equally probable arrangement. So now when these two are pulled this way and these two are pulled this way, ultimately we end up with gametes that have one copy of chromosome 1 from your mom and a copy of chromosome 2 from your dad, compared to this one, which had both from your mom. And this is chromosome 1 from your dad and chromosome 2 from your mom. So this created 4 possible combinations of chromosomes. So if with a cell with 2 chromosomes, that's not very many. 4 total possible combinations. This is all the ways the chromosomes could line up. But the formula for this is equal to 2 to the n, where n is the number of homologous chromosomes and this is equal to the total number of possibilities so if your n is equal to 2, the number of possible different types of gametes you can make is 4. If your n is 23, 2 to the 23rd, then you have 8,388,608 different possible combinations. Okay, so just with the way that the chromosomes that you got from your mom and your dad are lining up in metaphase here, and then how they're being pulled apart in anaphase, gives you over 800 million different possible gametes, eggs and sperm that you can create. And then when you also add in the fact that all of the chromosomes are experiencing crossing over, so they're exchanging genetic information, the possibility that you will create 2 either eggs or sperm that are exactly the same goes down to a very small percentage. So basically, every single gamete that you make is a unique combination of alleles, which is pretty amazing. Okay, so in meiosis, next thing that happens is that in anaphase, instead of sister chromatids separating like they do in mitosis, homologous chromosomes are pulled apart. So already, by the time we get to telophase, we have reduced the number of chromosomes in these 2 resulting cells by half. Because remember, this was chromosome number 1 that you got from your mom, and this is chromosome number 1 that you got from your dad. I mean, not exactly, because they had experienced crossing over, so now they're mixed and matched up. But when they separated, now this resulting cell only has 1 of chromosome number 1, and this resulting cell only has 1 of chromosome number 1 as well, as compared to this resulting cell has 2 copies of chromosome number 1. So we have accomplished our goal already of creating variation and cutting the genetic information in half. But we still have one more round of meiosis to go through because these sister chromatids need to separate. So meiosis 2, we undergo without replicating the DNA again, we undergo another round of prophase and then metaphase where the chromosomes are lined up. And these aren't really true sister chromatids anymore because they underwent crossing over. So you can see that they're kind of like a checkerboard pattern or a mismatch. So they're not true clones, but they separate again during anaphase and then telophase. And ultimately, we create 4 cells, and each of these cells is different than the original cell that they came from, and also it has half the genetic information. So these resulting cells have 2 copies. This is chromosome number 1, and this is chromosome number 1. This is chromosome number 2, this is chromosome number 2. This cell only has 1 copy of chromosome number 1 and 1 copy of chromosome number 2. And then the same is true for this cell. 1 copy of chromosome number 1, 1 copy of chromosome number 2. So we have accomplished our goal of creating variation and cutting the genetic information in half. Okay, that's all. Bye.